So we'll finish up the heart chapter this week, today and on Thursday, and it will be some information that you'll be able to use during your heart eval next week. So notes are up for your heart eval. There's some guidelines that are posted for you, so make sure you review that and make sure you rehearse, because you'd be surprised how fast 2 minutes go by. You have 2 minutes to do this. It's worth 10 points, and there is a sequence that if you follow it, you don't have to be repetitive. So remember, if you've already said it, do you need to say it again? No. So make it simple, simplify it, and that will make it easier for you to garner the most points. Now back to our heart that we have here for the cadaveric view. Posterior is right here, and it's easy to actually see our pulmonary arteries, which are taking blood away from the heart, going off to the right side of the heart, and also the left side of the heart to the right lungs, the left lungs. So those arteries are taking oxygen poor blood to the lungs for oxygenation, and the blood will come back to the heart, to the other side, to the left side of the heart, via veins. So veins are always taking blood back to the heart. So the pulmonary veins are going to be responsible for that. And in our specimens, our pulmonary veins are pretty much non-existent. It's a large hole in some of the specimens, but there are still some holes in other specimens. If you've already said the apex of the heart from the anterior aspect, you don't have to say it from the posterior aspect, things along that line. Coronary sinus, that is in a atrioventricular sulcus or groove posteriorly and empties into the right side of the heart. Again, the inferior vena cava is a very short structure on the human heart because the heart literally sits on the diaphragm, and so it's hard to actually cut through. This is saying superior vena cava, but you know it's kind of hard to tell from this posterior view, correct? So you have to realize that the picture is giving you some ideas where to find them, but that view is not the best view for actually seeing those structures. Let's talk about the pericardium. Most of your specimens do not have a pericardial sac associated with them. So let's go ahead and go over the pericardial sac so you realize that that was actually a specimen that was not obtained in a way that would give you the satisfaction of understanding how the pericardial sac is applied to the heart. Our notes say the heart is enclosed in a sac with 3 distinct layers. The pericardial sac just has 2 layers. The 2 layers of the pericardial sac, like a grocery sac, people, is the outer aspect of the sac and then the inner aspect of the sac. The outer aspect of the sac is your fibrous pericardium, and it's made out of dense connective tissue. Is that going to be dense regular or dense irregular, CT? Irregular, because we don't want it to split. Now, going inside the sac, it is lined with a serous membrane. And we've talked about serous membranes before when we talked about ventral body cavities. And the lining is always parietal. So inside the sac is the parietal pericardium. It's nice, slick, and shiny. And then this serous membrane also covers the organ in the cavity in which you find that. And so that's the third aspect of this pericardium, the visceral pericardium. So the serous membrane covers the heart, which is suspended inside the pericardial sac. And our notes state it's continuous with the parietal at the base of the heart, which is where the great vessels actually come in and out and where our atria are actually located. Our cartoon here is showing you the fibrous pericardium on the outside and on the outside, and then it's showing you the nice thin layer of parietal pericardium or serosa. And then on the surface of the heart, we have that visceral layer. And between the serous pericardium, the parietal and the visceral, we have a cavity, a space. And this space is actually truly really small. And the material that's in this space is serous fluid. serous fluid. It's a serous membrane. It produces serous fluid. It's clear and it's there to decrease friction between the inner aspect of the sac and the surface of the heart. Because we don't want to have the heart, which is actively beating, to actually rub up against the inner surface of that sac and stick. And so we have that nice serous fluid in that pericardial cavity. So let's put a few notes about the pericardial cavity. It's a very small space between the parietal and visceral pericardium. Contains a small amount of serous fluid that is designed to decrease friction between the two surfaces. If you have too much fluid in that area, if we actually had for some reason a bleed into that pericardial cavity, how would that impact the movement of the heart muscle itself? It would be very constrictive, which would be incompatible actually with life. And there are situations where that actually occurs, and that's called cardiac tamponade. You don't have to know that right now, but later on in your careers you will experience somebody who unfortunately has cardiac tamponade. Now, what about if you don't have that serous fluid there? What if there's an inflammatory process that actually decreases the serous fluid and actually causes collagen cross-bridging between the two serous layers? That's pericarditis. It's an inflammatory process, and that is going to be also restricted of the movement of the heart, but nowhere near as restrictive as if you have it, the whole area filled up with actually fluid that actually will limit the heart movement. But because now we have little scar tissue back and forth between the two layers, we actually will have symptoms, and we'll have a lot of chest pain associated with that. It's not a heart attack, but it is definitely very painful. So pericarditis is actually telling you we have inflammation between those two serous layers. Looking at the heart wall itself, we also have three layers here. We have epicardium, which is the outermost layer, which we could also call visceral pericardium. So they're synonyms. Myocardium, it says fibrous connective tissue skeleton plus cardiac muscle. Well, you don't see the skeleton, all you see is the meat. So for you, myocardium looks just like a thick muscular layer. But realize those muscle cells are actually scaffolded in their location. So myocardium, thickest, no matter where you are, whether it's atria or ventricles. Endocardium, that's inside, and it lines the chambers, our atrial chambers, as well as covers the valves, and also lines our ventricular chambers. So now let's take a look at the various chambers, of which there are just two types, a total of 4 chambers, and the great vessels, of which there are also just 2 types. The blood vessels that we'll be looking at are arteries and veins, and arteries always take blood away from the heart, and veins always take blood back to the heart. Arteries also always come off of ventricles, and veins always empty into atria. Taking a look at the chambers, we kind of say 2 upper and 2 lower. And that's because it's kind of how people like to view the heart. The heart doesn't actually, it's more at an angle, but still we can say our atria are superior to our ventricles. So we're going to talk about the 2 upper first, which are the atria. Indeed, they possess thin walls, and that is because they are considered low pressure receiving chambers. and they are designed for some distensibility. When we take a look at them, what everybody notices, because it's hard to actually see the atrial walls, is we notice the auricles. They are like our auricles of our own ears, or pinna, our ear-like flaps that are found with both atria. So the parts that we actually see here, these that we're listing, are associated with both atria. So in the auricles are these lovely muscles, these pectinate muscles that actually line the auricles. The auricles themselves are the only really obvious muscular component of our atria. There is some muscle in the other aspects of the atria, but it's not really obvious to us. So the auricles are the muscular components of the atria, and when we take a look inside the auricles, we see those muscles, the pectinate muscles. The other thing about the auricles provide a little extra area to our atria. And so I'll say volume to our atria. The fossa ovalis is an oval depression in the interatrial septum, and it is a remnant of the foramen ovale. And this foramen ovale is a fetal shunt. We have a couple of different ways that we will keep blood going, keep it from going to the lungs for oxygenation in the fetus, and the the foramen ovale is actually one of those. The fossa ovalis is in, I already have it here, in the interatrial septum, and it's in the posterior aspect. Of our interatrial septum. So those are things in common to both atria. So now let's take a look at some of the things that cause differences in our two atria. The right atrium is going to receive used blood. Used blood is oxygen poor, carbon dioxide rich. That is what we mean by used blood. And the veins that are going to empty into the right atrium, because that's what all these are veins. In other words, venous blood, because they're bringing blood back to the heart, are the superior vena cava, which is bringing used blood to the heart that originates from above the heart. The inferior vena cava, which is bringing used blood to the heart from below the heart. And our coronary sinus, which is bringing used blood to the heart that comes from the heart muscle itself. Even though the heart has all this blood inside of it, that's not the blood that feeds the heart. The blood that feeds the heart has to go through its own circulatory path to where it gets into capillaries. And so we actually have our own separate circulation for the heart. And so when the heart is done with its blood and it's sending it back, or when the myocardium is done with the capillaries, the nutrient transfer, it's going to go into the veins, and eventually all of that blood will empty into the coronary sinus, which goes into the right atrium. In our left atrium, again, veins are going to actually enter into the left atrium, and we state that it contains fresh blood from the lungs. So that's oxygen-rich carbon dioxide poor blood. When we take a look at the ventricles and compare the thickness of their walls to the atrial walls, absolutely they have thick walls in comparison. So relatively thick walls, especially when you compare them to the atria. Things that we actually see in both the right and the left ventricles are the interventricular septum when you're on the inside. On the inside also you see trabeculae carneae, that is actually the ridged appearance to the inner lining of our ventricles. And also you will see papillary muscles. And those are protrusions from the ventricular wall that have chordae tendineae attached to them, which are then attached to the valve leaflets. And so those are also structures that we find on both sides. When we take a look at the right ventricle and compare it to the left ventricle, its wall is thin in comparison to the left. And that's right ventricle will have an artery leaving that, and that artery is called the pulmonary trunk. And that is going to send used blood to our lungs for oxygenation. Our left ventricle indeed has exceedingly thick walls. It's easy to figure out right from the left because of such degree of thickness of that left ventricular wall. The artery leaving the left ventricle is the aorta, and the aorta is going to take fresh blood to the entire body except the lungs. Because the pulmonary trunk takes blood to the lungs. Inside the pulmonary trunk and the aorta are some valves, and our valves are designed to make sure blood travels through the heart in only one direction. So we have valves between our atria and ventricles, as well as valves between the ventricles and the arteries that actually exit those ventricles. Let's take a look at the valves that are designed to keep our blood flowing in only one direction, in atria, out ventricles. The first valves that we encounter are called the AV valves, or atrioventricular valves, and that's a generic term. It's just like saying gonads is a generic term for testes and ovaries. But if you have a male cat, you won't say gonads, you'll say testes whenever you're actually referring to those, and vice versa. So the AV valves is a generic term, and now we're going to be specific as to which side of the heart that we are on. If we are on the right side of the heart, they're called tricuspid valves. If we are on the left side of the heart, they're called bicuspid valves or mitral valves. And tricuspid means 3 leaflets to them. Bicuspid or mitral, 2 leaflets to them. The atrioventricular valves have coming off of them chordae tendineae. Your book calls them another name, correct? Your lab manual? Tendinous cords. So tendinous cords are also acceptable, but we've been using chordae tendineae for a long time, and so we're going to keep that terminology. So off the valves, there's an attachment, some lines, so to speak, chordae tendineae, and they are then embedded into the papillary muscle. So the papillary muscle is myocardium that is actually sticking out from the heart muscle wall on the inside. And so we see in our picture here, this is referring to our papillary muscle. Here are the chordae tendineae, and here is a cusp or leaflet. Now, this area is the atrium, which is superior to the ventricles, and you know this is the ventricle and that is the atrium because you can see the trabeculae carneae in the ventricular picture, correct? So there's trabeculae carneae, there's this interesting internal pattern to the muscle. And blood is going to flow from the atria into the ventricle, and it does that, we'll talk about that a little bit more, via mostly a passive process. But the atria will eventually contract when it's time to force the last little bit of blood that they have into the ventricles. Then when the ventricle contracts, you don't want the blood going back into the atria, correct? You want it going out. If we're in the left, we want it going out the aorta. If we're in the right, we want it going out the pulmonary trunk. And so in order for that to happen, we have to close those AV valves, no matter which ventricle we're in. And what happens is the papillary muscles will actually contract. And at first that seems pretty strange to you because that would actually make you think it's going to keep it open, correct? Okay. What I want you to realize is the heart doesn't, the ventricles don't contract like this. The ventricles contract like this. So when the heart muscle contracts, I want you to actually visualize, because this is also internet people listen to this too. I want you to visualize that the apex of the heart is actually moving up towards the AV valves, and it's twisting. And because it's moving up towards the AV valves and twisting, those, if we don't put a little bit of shoring up on those chordae tendineae, those valves will prolapse. You guys follow me? So that's why it looks a little strange at first to actually realize papillary muscles shorten. But if you think of the bottom of the heart coming up towards those valves, if you don't start to actually shorten the muscle and therefore pull on those valves leaflets, then those leaflets will actually prolapse and give you regurgitation or backward flow into the atria. So that's a very different mechanism of those valves than the semilunars. Yes?
So that the prolapse might involve in that the regurgitation of the blood flow. Does the blood flow get stuck up in that atria, or does it,
well,
how does it flow normally?
If the valves are not closing properly, so if they don't close and allow no passage of blood back into the atria, that is called prolapse. So there's a leaflet or a flap that is actually flowing into the atria, and with it, the blood. That typically is going to occur if you have a dysfunctional papillary muscle, correct? Or if you have some broken chordae tendineae, correct? Or if for some reason you have extra stretchy chordae tendineae. So if they're too stretchy, then that means that that back pressure of that blood forcing those flaps also to that closed position, then that means if you're too lax, then you can actually have regurgitation. Well, that blood will go in there into the atria, but the atria are again going to fill up, and we already know that when the atria fill up, they're eventually going to then fill up the ventricles. So what that means is you never have a complete emptying of the ventricular volume into its artery, correct? You get backup, so you get a little bit stolen from you whenever this actually occurs, correct? And so we're going to talk about this too in a little bit, because I'm going to ask you, I want you to think about this even before we get to this. Is it reasonable for the right side of the heart and the left side of the heart to pump the same volume of blood? If you do not pump the same volume of blood, let me ask you, who's pumping more blood than the other side? So if the left side pumps more blood, where is it getting its blood from? Ultimately the right. So then you're stealing from the left, correct? If you're saying that the right and the left side do not pump the same volume, remember the left side of the heart is getting its blood ultimately from the right side of the heart. Yeah. So even though they look different intravascular volume-wise to you, you have lumped up, you have two beats, and that's not right ventricle, left ventricle that you actually are hearing. They both contract at the same time. They both eject their blood at the same time. If you have one ejecting more blood than the other, ultimately you're going to have a problem. So they both eject the same volume of blood, which also goes to show you how absolutely rich the capillary beds are of your lungs. So your lungs are a sponge, and you guys know sponges can hold a large volume of water, can they not? And so that's one of the things that it's conceptually at first difficult to actually understand that, but if you actually think of flow dynamics, if they don't pump the same volume, ultimately something's going to be problematic and not very long. But those are the AV valves. They have a very unique construction. So we have the cusp or leaflet, we have the chordae tendineae or tendinous cords, and we have the papillary muscle. They all have to be functioning properly in order to get proper closure when the ventricles contract. You guys follow me with that? Okay, so when the ventricles contract, the blood is supposed to go out the artery from that ventricle, pulmonary trunk for right, aorta for left. So let's take a look now at the construction of the valves at the base of the aorta and the base of the pulmonary trunk. They're called semilunar valves because they look like half moons to people when they first actually discovered them. So our notes say at the entrance of the aorta and the pulmonary trunk. Well, this means the inside, correct? Because they're not on the outside of the heart. We're dealing with blood flow inside the heart. So take a look at our picture here. It says semilunar valve open. And the leaflets are up against the inner walls of our pulmonary trunk or aorta. When the semilunar valves are closed, that means the blood's not supposed to flow in the other direction. And so we see here the semilunar valves are closed. And their construction is really cool. Even though they have no chordae tendineae, and we have no papillary muscles, and we have a pocket-like shape. That prevents backflow or regurgitation again. And so today I happen to have on this nice little vest, and it's got some pockets in this. And the pockets in my vest are like semilunar valves. And the pockets in my pants are like semilunar valves. So I'm pretending my legs are the ventricles, and they're contracting and they're sending blood bouche superiorly, right? Isn't that kind of how we think of it? Okay. So the blood's going upwards and it's going over the outer aspect of the, or the luminal aspect of the semilunar valves, and it pushes them up against the wall, correct? Now, when the ventricles stop contracting, what does the blood want to do? Wants to fall back into the ventricles, correct? And there's many reasons for that. We'll get into that a little bit later, but it does want to fall back into the ventricles. And so indeed, as my blood falls back, if I can get my hands in the right spot, as my blood falls back into the ventricles, it falls into these semilunar valves, causing them to billow out and therefore close. So they're like shirt pockets or pants pockets, these semilunar valves, and we have 3 of them on each side, and so they'll all come together and they'll prevent backflow. Totally different construction, but still effective in providing one-way circulation. So the backflow causes them to close. And when they close, the blood bounces off of them and goes back the way it's supposed to go. You guys follow that? It's like a bouncing ball. It went down, but then it caused the, quote, closure of the semilunar valves, and bounces back and goes the way it's supposed to go. So the aortic semilunar valves indeed are at the base of the aorta, and they contain coronary ostia. How many coronary ostia do we have? We have 2 coronary ostia. We have right leading to the right coronary artery, and we have the left leading to the left coronary artery. How many cusps do we have? We have 3. So one of them is actually empty, has nothing in there. So the pulmonary semilunar valves are going to control the flow into the pulmonary trunk. So these valves control the flow into whatever valve, into whatever artery they're associated with. Control the flow into pulmonary trunk. We have 3 flaps and 3 flaps, or cusps. And controls flow into aorta. Now, what if they become scarred? What will the scarring do to their efficiency and function? If they become scarred, how easy will it be for the blood to leave the ventricle first? Difficult. And then because they're scarred, how easy will they actually come together to close properly? Also difficult. So that's called stenosis. So scarring of our cusps or leaflets will actually be a stenotic process. It will cause difficulty in getting blood out, and it will cause blood to regurgitate or backflow in, which will then even make it more difficult or require the ventricles to work harder, correct? So stenosis is difficult, and also what if something happened and one of those valves ruptured? It would be easy for the blood to leave, correct? But then we would also have regurgitation that would actually occur. So there's all kinds of reasons for actual backward blood flow, and the reasons are always going to be related to valve failure. The valves are failing to do their job, which is actually to prevent backflow. So realize the aortic, all of our semilunar valves are like pockets, pocket-like shape. Let's take a look at coronary circulation, because you guys are working on that lab, dissecting out the coronary blood vessels. So blood flow to the heart tissue itself, to the actual myocardium. So we have coronary arteries, which are going to exit the aorta via their ostia, which there are just 2, even though there are 3 valve leaflets. And then coronary veins collecting used blood and returning this to the heart via the coronary sinus. So what side of the heart does used blood always go to? The right side. So returning it to the right atrium, correct? Arteries will eventually give you capillaries, which are going to feed the heart, which will then eventually give you veins, which will eventually return back to the heart via the coronary sinus. What's important, this is our right coronary artery. It's coming out of the aorta, but snakes around the pulmonary trunk, and then is found easily in the atrioventricular groove, or sulcus, and you follow it all the way around to the back side of the heart, and it will give rise to the posterior interventricular coronary artery, correct? In the human, we also have a marginal artery. So we have a right marginal artery. That is the inferior margin of the heart, where that actually feeds that area. So the right is feeding, I hope you realize, the right ventricle as well as the right atrium, because it's actually in that little sulcus. It's also going to feed the muscle of the right atrium. The left coronary artery, which is kind of ghosted out in this picture, is going to come out behind the pulmonary trunk onto the left side of the heart. One of the things that you have to realize is this picture, when you take a close look at it, makes it look like the left coronary artery is about a couple of inches long. And I want to tell you, you're lucky if it's a centimeter long. So the right coronary artery is a centimeter or less in its length, and it will immediately branch into our anterior interventricular coronary artery or our circumflex coronary artery. And this picture really does give it no justice because it's exceedingly short and it makes it look like it's long here. So the circumflex coronary artery, I hope you realize, is actually in the left atrial ventricular sulcus or groove. And that's how you find it, and it's going to circle on around. That's why it's called circumflex, circumferential, circles on around, and it circles on around and feeds the left ventricle and the left atria and the bulk of the posterior aspect of the left ventricle too. The interventricular artery, it's going to feed predominantly the left ventricle, but we'll also have some bleed over to the right ventricle, correct, from the anterior aspect. Coronary veins taking now the used blood, so the heart used that blood, and now we need to return it back into the right atrium, and on the anterior aspect of the heart, adjacent to our anterior interventricular coronary artery, is the great cardiac vein. It will come up and actually go over the circumflex artery and go into the atrioventricular sulcus or groove on the left side. Now this is a vein, and one of the things I want you to realize, I want you to always remember, arteries are muscular and veins are flabby. And the circumflex vein has to travel over the artery because if it traveled under the artery, what would happen to it by the weight of the artery itself? It would be almost like a tourniquet, correct? So veins, as you are going to discover in your dissection work, are typically superficial to the arteries. Veins don't like to be under an artery. They like to be superficial to the artery because the artery itself would act like a tourniquet and actually impede venous return. So the circumflex artery is underneath our great cardiac vein as it goes in that sulcus and then to the back side of the heart where it will empty into our coronary sinus. We have a small cardiac vein which is on the margin of the heart, found with the right marginal artery, and our middle cardiac vein is found with our posterior interventricular coronary artery. All of those guys actually empty into the coronary sinus.
And that's on the posterior side, right? In the middle.
Yeah, that's on the posterior side. And so these are actually better photos than our old texts and lab manuals actually had. So they ghost out a little bit better, I think, for posterior aspect. Now, I want to ask you a question about circulation to the heart muscle itself. Remember where the coronary ostia are? That's the beginning of the blood flow to the heart. You guys understand that now. So when the left ventricle contracts, what do we know about the coronary ostia's opening? It's obstructed, correct? It is closed off by the flaps. Because remember, the flaps are up against the wall because the blood is whooshing on by, pushing them against the wall. So when the ventricle is contracting, it is not receiving any coronary arterial oxygenated blood flow, correct? Now, at first that seems like that's not right. But let me ask you, as you clench your fist, what is better to infuse the blood into the muscle of your fist, a clenched fist or a relaxed fist? So this is actually a very slick trick. The heart muscle is working and it's all firm and solid, making it hard to perfuse it, to actually feed it blood. And so the ostia, or the openings, are closed off. So blood is not flowing into the heart muscle when it's a clenched fist, so to speak. As soon as it relaxes, we know that the blood is going to fall back into the cusps, and the cusps are going to close, but when they close, what do we know about the ostia now? They are now open, and remember that blood is falling down with some force, because as we already kind of figured out, the left ventricle is such a big muscle because it's sending blood a long way off. Okay, so it's falling down with a lot of force, and it literally bounces and it just pushes blood into the coronary ostia, squirts it into our right and left coronary arteries. So that's actually pretty slick too, if you think about that. So those ostia and their location are actually ideal for proper feeding of the heart. So the heart feeds when it's relaxed. And the other thing, as you guys have noticed about the veins are kind of embedded in the heart muscle itself, correct? And so what that means is, if the veins are embedded in the heart muscle itself, that means when the heart muscle contracts, it literally squirts the venous blood along. You guys follow that too? So it's a pretty slick way to actually take care of the heart's needs. And so that's why whenever there's any restriction of coronary blood vessels, that means, like when people get angina, that means that what blood flow we're getting when we're relaxed isn't taking care of our needs. But the heart, if it's got adequate open vessels, is actually the blood flow to the heart muscle itself is excellent for taking care of the heart's needs. Pathway of blood through the heart. Two main circuits, the pulmonary circuit and the systemic circuit. Pulmonary circuit means we're taking used blood, oxygen poor, carbon dioxide rich, from the body to the lungs and then back to the heart again. And the function is to reoxygenate blood. And we'll take this from the right side of the heart. So this is the right side of the heart. And we'll send it to the lungs. And then to the left side of the heart. But the pulmonary circuit is considered the right pump. And is often called the right circuit because of that. The systemic circuit is now taking that fresh blood that it got from the lungs to all parts of the body and then back again to then go to the right side of the heart for reoxygenation. And again, we call this the left circuit or pump. So we have two cartoons here showing you pulmonary circulation, the other showing you systemic circulation. And it's a good idea to take a close look at them, but we're going to draw our own cartoon model of pulmonary circulation and systemic circulation. So in this space here, let's put some lungs in the middle. Remember, you don't have to be an artist to be an A&P. You just have to make your drawings look like somebody can recognize them. So here are the lungs. So we have two sides to our lungs with that. And I'm going to make a box over here, a rectangular box. And I'm going to make the top a little shorter than the bottom. And I'm going to do the same thing on the other side. And make another box. Rectangular one. And I'm going to have connection between my lungs and an atrium. And I have two lines. One is meant to be from the right side of the lungs and the other is meant to be from the left side of the lungs. And if you want to draw yours a little bit more that way to make you happy, that's great. From the other side, I'm going to make a connection to my lungs over here from the ventricle, because this is indeed going to be our atria, and this is going to be our ventricle. I'm going to have you tell me soon which one is the right and which one is the left, based upon my drawings so far. So right now, which is the right pump and which is the left pump? So this guy over here is the right, correct? Because it's sending, the ventricle is sending blood to the lungs. So this is PT for pulmonary trunk, correct? So this is then the left side. And these would be PVs, right? Pulmonary veins, correct? Because veins always go into atria and arteries always leave via the ventricles. So now, what's the vessel that leaves the left ventricle?
Aorta.
Aorta. So let's draw our aorta going to our body. And this is how I'm going to draw my body. And then from the body, blood is going to go back to what chamber? Right atrium. So we have to be a little creative over here. So I'm going to go from this area up here. And then I'm going to go from this area I'm going to have a little overlap, but I think you guys can appreciate what I'm trying to do here. One of these is the SVC and one of these is the IVC, correct? So the one from the head is the SVC and the one from the body is the IVC. What is the valve between the right atrium and ventricle called? Tricuspid. So let's put a little T here. And the other one is called bicuspid or mitral. And so now we're going to talk a little bit about circulatory dynamics a little bit before we even get into this. If for some reason we have a problem with our bicuspid valve, so bicuspid problem with prolapse. What does prolapse mean to you guys? One of the leaflets is going into the atria when the ventricles are contracting, correct? So that means that the ventricle is sending some of its blood to the atria and not all of its blood is actually going to the body, correct? All right, so the body's getting a little bit robbed and the atria is getting a little bit of backflow. If the atria is getting backflow, what's going to happen to the, so they're not emptying enough, what's going to happen to the pressure in our pulmonary veins? The pressure is going to go up, okay? So PV pressure, let's erase that little guy there. So this means pulmonary vein pressure increases. Well, if pulmonary vein pressure increases, this means that the capillaries are going to be under increased pressure in the lungs, correct? And when capillaries, I'll let you know, when you have increased pressure of the capillaries from their luminal aspect, you get leaky capillaries. Can you guys follow me with that? Capillaries are very delicate structures and they're not designed to handle backup. They don't back up well, they leak. And when these leak, if we have increased pulmonary vein pressure, we end up with leaky lung capillaries. And when you listen to their lungs, what do you hear?
It has to be.
You hear crackle, crackle, crackle. Lung edema or fluid in your lungs. So that's with a bicuspid valve prolapse problem. It causes backup. Now, what if there was stenosis of that bicuspid valve? Stenosis means almost the same thing as prolapse, correct? Because if the bicuspid valve or the mitral valve is stenotic, how easy is it for you to empty the atria? It's difficult. So again, we get backup pressure, and as long as you get backup pressure, the backup will be experienced most profoundly in the capillaries that are actually precluding or in front of the area where the pressure is backing up. So let's take a look at edema of the legs, right? Don't you see edema of the legs all the time in a lot of people? So let's take a look at that process there too. So now we're talking about a T valve, tricuspid valve. So let's go ahead and because we did prolapse, let's do stenosis. So that means that the right atria do not empty completely, and the right ventricle is getting a little shorted, so to speak, on the amount of blood it's going to send off to the lungs. Well, when the right atrial pressure, so then our atrial increases, that is going to cause an increase in inferior vena cava pressure. And the reason I say the inferior vena cava pressure is going to increase because it's gravity, people. We're going to have more of the backup in the area where gravity is playing a bigger role. So the inferior vena cava pressure will increase, and therefore you will have leg capillaries becoming leaky, which gives you leg edema.
So what do you get when you have pitting edema? What is and it just doesn't rebound as well as just normal?
Pitting edema is the same thing as leg edema. Any sort of fluid in the extremities is edema. Even fluid in the lungs is edema, but what do we call that? What do we call that when we have right heart failure, left heart failure? What do we call that edema in the lungs?
Congestive heart failure.
Indeed, congestive heart failure. So congestive heart failure is a left pump issue, is it not? So the right pump issue is going to be actually experienced in our extremities, and more likely our extremities that are subject to gravity. So these are actually some problems you're supposed to work through when actually you do your prep for the upcoming exam for this, because there'll be questions related to that. Now we're into cardiac physiology, and we're going to start with histology, our microscopic anatomy, and indeed myocardiocytes or cardiomyocytes, you can call them either one, are short rectangular cells with a single nucleus, totally different from our skeletal muscle cells, which are long with multinucleated. They are, however, striated, correct? They're lightly striated, so we still have the sarcomere situation with these guys like we do skeletal muscle cells. The interesting thing about heart muscle cells, and so here we also have that little drawing of sarcomere to let you, to remind you of that. We have this phenomenon called intercalated discs. They're very obvious whenever we have a good specimen that has proper staining. So those guys are intercalated discs. Those are the ends of a heart muscle cell. In the intercalated disc, what makes them up is this weird, wavy, undulated, or papillary kind of pattern with desmosomes, which are tennis shoelace kind of things, holding things together, and gap junctions. gap junctions, remember, are almost like one cell's connected to the other cell. They're bridging each other. So those gap junctions allow, whenever one cell experiences an electrical event, that electrical event will be passed on to the next cell, which will be passed on to the next cell. So our intercalated discs are indeed a combination of this undulating membrane held together with desmosomes and gap junctions. As our notes state here, the desmosomes are spot welding, or there for strength. And the gap junctions are going to allow ions to flow from one cell to the next, enhancing the transmission of an electrical impulse. And what's important about this, with these gap junctions, is I have, right here in this picture, I have 3 heart muscle cells. And because of the gap junctions, these 3 heart muscle cells all act as one. And so we have these short cells, and the heart actually acts like it's a single unit as far as electrical impulse and contraction, even though it's made up of these very distinct, actually small heart muscle cells. So what happens here when we have ions flowing in here, it will be felt here, and then it will be actually transmitted to the next cell. So they all will actually have a wave of contraction running down the actual length of this track of cells. Another thing about heart muscle cells, they have indeed many mitochondria, more numerous than skeletal muscle cells. And our notes here talk about differences between skeletal and cardiac contraction, all or none. Remember, we talked about a skeletal muscle cell is an all or none contractile process. The cell itself contracts or it doesn't. Well, what we're talking about here with all or none is not a cell. We're talking about the entire heart contracts as a unit. So when one cell starts its contraction wave, all of them will actually go ahead and contract, and the heart contracts as a unit. The other thing about the heart, it has automaticity, which is interesting. The heart was born to beat. It was born to pump blood. And it's because we have some very important cardiac muscle cells. They're specialized to where they can actually initiate their own depolarization. You don't have to tell them to depolarize. They will depolarize on their own, and these are our pacemaker cells, or pacer cells. And we'll get into those in just a moment. The other thing about heart muscle cells is that heart muscle cells have a long refractory period. A refractory period means we cannot contract again. So we have an action potential, and we have this time where we cannot generate another action potential, which means we cannot contract again. So remember, action potentials are always associated with contractions, right? When we talked about skeletal muscle world, action potential in cardiac muscle world is always associated with a cardiac muscle contraction. Well, a refractory period means we can't generate another action potential and we cannot contract. So 250 versus 1 or 2 milliseconds. 250 is for cardiac and 1 to 2 milliseconds for skeletal muscle. This is important because we don't want the heart muscle to go into tetany. So this prevents tetany. And also provides proper filling of the heart. the faster your heart beats, the less time your heart has for proper filling. The slower your heart beats, and that's another reason the athletes' hearts are so nice and slow in their heart rate, is that we have really great time for filling, and then we have maximal ejection of this large volume of blood to actually go ahead and supply your muscles. So the longer you have for your time in between heartbeats, the more blood you can put in. Can you guys follow that? And so that's a very important thing too about this prolonged refractory period. The other thing that's not up here in the notes that I'm going to add to the notes for this is that heart muscle cells have lots of myoglobin. Do you know what myoglobin is for? It's for oxygen attachment to this protein inside your cells. And it makes a lot of sense that heart muscle has a lot of myoglobin because when the heart is working, you know it is not getting oxygenated, correct? And so the myoglobin inside the heart muscle cells is always what I call the oxygen bank. And so when the heart muscle cell is working really hard, it takes from the oxygen bank, and we replete or replace the oxygen bank whenever we have a relaxation of the heart muscle. Can you guys follow that aspect? So myoglobin is a very important protein for heart muscle cells to actually help them function properly when they're working. Now, let's take a look at the mechanisms of contraction, which is always associated with an action potential. Remember, action potentials are very similar for all excitable tissues. APs, nervous tissue, and muscle tissue, for example. Our notes here state action potential hits a muscle cell, muscle cells, and when the action potential hits a muscle cell, it causes the cell's sodium gates to open, and you know sodium's going to flood into the cell. So this is actually just a regular heart muscle cell. What's interesting is the sodium gates stay open until a peak of our action potential. And what was that for when we did nervous tissue? Our total action potential spike was 100 millivolts, and so we went up to 30, positive 30 millivolts, correct? Okay, so sodium gates open, and when we hit our positive 30 millivolts, the sodium gates close. And we know what causes potassium gates to open, correct? Isn't it that 30 millivolt spike? So potassium gates open. But also, you guys remember at the end of the axon, at the axon endings or terminals, whenever the action potential gets down there, that 30 millivolts gets down there. Do you guys remember what also goes into the axonal terminals before exocytosis can occur? Calcium entry. That's voltage sensitive. Okay, so this is really interesting because all of a sudden at this peak, when sodium gates are closing, potassium gates are opening, all of a sudden we have calcium gates opening. And when these calcium gates open, they are going to cause a plateau. So the plateau is something that we have not seen with action potentials before at all. So we're going to see a plateau. And the calcium gates are actually then going to close because the plateau is not flat. It's got a little bit of a decline to it. Calcium gates will close. And because we're still up there, our potassium gates are actually remaining open until we actually reestablish our resting membrane potential. And then those potassium gates will actually close. So adding to our notes for A, sodium gates open, and this depolarizes the cell. So this is a heart muscle cell. And then potassium gates close, sodium gates close, potassium gates open, and we mentioned due to reaching positive 30 millivolts. Calcium gates also open because of this positive 30 millivolts. Because calcium gates are voltage sensitive. They are, interestingly, they are slow to open. And that slowness to open is part of what causes this plateau also. So the arrow is going to be referring to that plateau. When our calcium gates finally close, this will absolutely stop our depolarization. And our potassium gates indeed will continue to stay open until we get back to our minus 50 or minus 55 for our resting membrane potential. So this is what it looks like. This is what a heart muscle cell action potential looks like. RMP, resting membrane potential. Sodium gates open and we get a nice upward spike just like we did with other action potentials. So RMP, resting membrane potential. Let's say up here is positive 30 millivolts. So we get to our positive 30 millivolts and our sodium gates close, potassium gates open, and we get this little spike, but then all of a sudden calcium gates are opened. Calcium gates close, and then we get all the way back to our resting membrane potential, and we do it all over again. So this is A, and let's see here. This is B. This is C. This spot here is D. And then this is E. That's an action potential for a heart muscle cell. This is not one for a pacer, and we're going to take a look at a pacer in just a little bit. Excitation and electrical events is referring to our pacer or nodal system. Our notes already down here have nodal system for you. And we have an intrinsic control of our heart rate. Our hearts are designed to beat at 100 beats a minute, period. So if you left them alone, if you didn't give them any sort of A&S input at all, your heart would beat at 100 beats a minute. Now, when you're in rest digest mode, you don't need to be beating at 100 beats a minute. So the parasympathetic nervous system, as we already realized, tells the heart to slow down. So the heart has got parasympathetic innervation and also has sympathetic innervation. Because then when we need fight, flight, 100 beats a minute may not be enough to take care of it, correct? And so the sympathetic is going to actually dictate that we need to increase our heart rate, and the heart will do that. So that's called the extrinsic control. Intrinsic means built into you. Do you guys remember the intrinsic muscles of the eye and the extrinsic muscles? So intrinsic means it's part of your construction built into you. Extrinsic means it's coming from an external source and being applied to you. That's what our intrinsic conduction system is all about. Cardiac cells, we're going to have some important specialized cardiac cells. acting as pacemakers. You guys have heard about pacemakers being implanted into people, and that's because the intrinsic conduction system has failed for that individual somewhere along the line. And we're going to take a look at that as we actually go through this. And I think that is a good place to stop. So we'll stop there and do a couple of clickers.